Hi, everyone. Thank you for being with us. Today we welcome you once more to a CE3C Encontro Ciência, and we are quite pleased to have today as our speaker Antonio Cordero, to whom I now thank in advance for being available to share his work with us. Thank you, Antonio. And Antonio, you can ask questions to Antonio directly at the end of the presentation. We have some time for that, but during the talk, if you have some questions, you can put it in the chat and we will read it at the end. And also I would like to say that the talk will be recorded, so if you don't want to appear, you can turn the video off, but then you can see it later in our YouTube channel. Antonio will be presented by Professor Solveig, to whom I thank also, and to whom I now give the floor. Thank you for you both. Thank you.
Thank you very much for those words, Patricia. So it's a pleasure for me to present Antonio Cordero, who started to worked in our group for a little while a few years ago, and now joined us with a Sec Junior in August 2021. So Antonio did his university studies in the US. So he graduated in Bachelor of Environmental Science in the Oregon State University, and then went on to do a PhD at Iowa State University in Ecology and Evolutionary Biology. He then did a postdoc in the University of Lund in Sweden between 2015 and 2017, where he focused on the evo-devo of turtle development. So he has worked most of his career with reptiles, but has now diverged to other organisms, and then he did in 2018 a 6-month project with us on an RNA-seq analysis of normal versus dystrophic fetal muscles in the mouse. And finally, he did a second postdoc in University of Tübingen, where he studied both the morphology and the physiology and the gene expression of the musculoskeletal system and the adaptations that occur in turtles, and also did some studies on lizard development. So it is a pleasure to present him here. I think he will talk a little bit about his past work and then kind of link it to what he's doing here with us in DEM, as well as with Ivo Cielo in the evolutionary ecology group. So it's a pleasure to hear this talk.
Thank you, Solveig, for the introduction. I will now share my screen. Okay.
Yeah. Yes. Perfect.
Yes. So my talk today is about development and evolution, and I will have some examples of how changes in the timing of developmental processes are relevant to evolution, and as well as biomedicine and even ecology. All right, so now I would like to define to you ontogeny and phylogeny. Ontogeny usually refers to the development of an organism, and we can break that down into different processes, which I will refer to as developmental processes in this talk. We have things like differentiation, which is a change in state, and this can be defined at the cell level or at the tissue level. There's also pattern formation, as well as growth, and also morphogenesis, which is change in shape. So, so for the purposes of this talk, I will refer to these processes. And they're very informative, of course, to patterns of evolution, of macroevolution. We have some examples here of ancestor-descendant relationships, which can be modeled by phylogenetic trees with phenotypes mapped onto the tree. We can see a scenario of trait loss, evolutionary innovation, changes in body size or trait size, as well as convergence. So the independent evolution of phenotypes. And today, actually, I have a couple of examples concerning size change and convergence. But first, I'm going to talk a little bit more about the theory behind ontogeny and phylogeny, let's say. Does ontogeny mirror phylogeny? So this is a classic question in evolutionary biology. And the answer is quite complicated. And yes and no. So in many cases, what we see in embryos does reflect what we see in adults. So it does reflect adult phenotypes. In other cases, not at all. But generally, what's expected is that the onset of ontogeny, the earlier stages of development, can be very variable, morphologically, as well as in the timing of developmental processes. But if you look at, for example, at vertebrates towards the middle of development, embryos tend to be quite similar to each other. They feature very similar traits. And the hypothesis is that this is because there's a peak in inductive interactions, developmental processes, and gene expression during this time. But then thereafter, the ontogenic trajectories of different lineages will diverge. And this is how differences in the adult phenotype are expected to be generated in development. Of course, there are many exceptions to this, which I cannot go into detail in the talk, but I just wanted to very briefly give you a very broad overview of the relationship between development and evolution. And I'll have some examples of this for you as well. We wanted to look at mammalian development. And so again, if we're looking at the adult phenotype, there are many differences. And to understand those differences, it's very important to kind of consider the entire life cycle of organisms. It's very important to look at trajectories, at ontogenic trajectories, if we want to find the mechanisms or changes in the processes that give rise to these differences at the adult stage. So this is more or less the framework of a lot of the work that I've done in the past and continue to do in the group at the Institute. And we'll give you an example of mammalian development. You can think of development as being split into two main phases. The first one concerns organ formation, so organogenesis, and that's what we're looking at in panel A of this figure. And then the second half of development, you could say, is really about growth. So we decided to sample all the information that was available for placental mammals, and we scored the timing of developmental processes in as many species as we could find. And so that's the phylogenetic tree of the species that were sampled in this exercise in panel B. And then in panel C, what we did was we computed interspecific variation in the timing of development. And what we found was that early in development, surprisingly, there was a lot of variation. And then you come to a period that we like to call the embryo to fetus transition, which is more or less kind of like in the middle of development, where mammalian placental mammals tend to be very similar in terms of the timing of developmental processes. And then there's a lot of divergence thereafter. And that's because the later stages of development is dominated by growth. So we found major differences, of course, in the growth period when comparing an elephant to a bat, for example. And then, of course, we also included human development, and domesticated species were very prominent in this sampling scheme. And so the main point here is that there is a point in development that tends to be very conserved, and mechanistically, that's it's very important to investigate. In slide, here we go. Yeah, and so here's the next slide. We wanted to kind of mechanistically explain what's going on, and this is an idea that others had pursued before, and we wanted to kind of like discuss it again. Earlier in the talk, I talked about different types of developmental processes, such as differentiation, pattern formation, morphogenesis, and growth. Early in development, you will observe all of these processes, and they're not mutually exclusive from one another. So they all depend on one another. And so all the errors that you're looking at in the early embryonic development box, you can think of them as being representative of interactions, so inductive interactions. Certain processes will induce changes in other processes. So this is happening all throughout development. So it's very dynamic. But what we find is that later on during that mid-developmental period, what's expected is that as organs begin to develop and differentiate, those inductive interactions also increase. And so this is what's been hypothesized to cause that high evolutionary conservation of the mid-developmental period. And then lastly, later in development, it's basically dominated by growth processes, as well as a bit of morphogenesis. And so we hypothesized that the late developmental stages are more susceptible to change and eventually evolutionary change. So this is kind of important for what we can expect for changes in developmental processes to produce in terms of phenotypic variation in animals. Okay, so there is some experimental evidence that that mid-developmental period is highly sensitive to mutations and possibly environmental perturbation. There have been very few studies, but one that induced mutations via applying a chemical agent during development of mice showed that fetuses and embryos tended to, they observed more fertility in embryos and fetuses during that mid-developmental period. And so this is actually one of the few studies that has produced experimental evidence of that sensitivity and why that period is so highly evolutionarily conserved, again, because of a high level of interactions, tissue-tissue interactions, genetic correlations. We can refer to them as well as genetic correlations. And so now I just want to show again that the framework for the talk, which is all about linking developmental processes with evolutionary patterns. I will have some examples of this in the talk from my own research. And so I will give you some examples of collaboration I did with some colleagues in Switzerland and Germany on chicken domestication, also on avian diversity in development. And the second part of the talk, I'll have some more examples on body size evolution, some ecologically relevant traits like limb length in lizards. And then to finish up the talk, I'll talk about some of the ongoing work in our group. And so on to the first part of the talk, evolution in human-controlled environments. So I was invited to work with colleagues at the University of Zurich on a really interesting project on the evolution of chicken under domestication. And this was a really fascinating project because we actually had the ancestral ontogeny, and we could compare it to the derived ontogenies in recently evolved domesticated lineages of the species. And so here we're showing the red jungle fowl, which is our ancestral state, the wild type, the white leghorn species, which is an egg layer. It's been bred for egg production. Then we have the broiler chicken, which is for meat production. And then we also sample a Japanese ornamental breed, as well as a fighting breed. And so what was really interesting about this project was that, as I mentioned earlier, was that we had ontogenies for ontogenetic trajectories for all of them. And so I thought that it was a really interesting opportunity to test the hypothesis that the genetic cost of domestication, whether it should lead to a reshuffling of the ancestral ontogenetic sequence in chicken, as well as growth pattern. And what I mean by the genetic cost of domestication is that it's expected that domesticated animals or any species undergoing human-mediated selection is more likely to accumulate genetic mutations and other changes in development might be permitted because the species are not being subjected to the natural selective regime. And so what we found was interesting because, well, before I tell you the findings, let me explain to you a little bit about what we expect generally. And so if you imagine that development is a sequence of events, which is shown here by the different colors, each color represents a different developmental event, each one associated with a different set of developmental processes. And so the ancestral sequence, which we can see in the top, generally will be maintained. Most of the sequences will be maintained especially early on in the derived lineages. And the different types of changes that we can expect to observe are terminal inversions, so very late in development, or additions to the sequence also occurring late in development, inversions late in development, or you could have the loss of some of these events. Very rare is to have a flip-flopping of the earlier steps in development, and also kind of rare to have added developmental processes to the ancestral sequence. So very generally and broadly speaking, this is some of the hypothetical scenarios that you could find, that you can predict in terms of how development could change across ontogeny. And so now I can show you some of the results. It turns out that interspecifically, so within Gallus chicken breeds, ontogenetic sequences were highly conserved. What I'm showing here is a ranked sequence of developmental events in chicken. So we described chicken embryos, and we recorded the timing of each developmental transformation. We had a set of about 100 to 120 character states, let's say, that we scored in chicken, in the wild type, I should say, pictured here in the x-axis, and then we simply compared to 4 breeds. And what we find is that there was very strong congruence in the sequence of developmental events. There were some variants. There was some flip-flopping, but we think this is due to, we could not really pin it down to a potential biological, the biological significance of some of the flip-flopping that we did observe in the sequence was very difficult to interpret, and we think it could be due to error. But generally, we would predict that development is highly evolutionarily conserved. So the sequence of developmental events, even during domestication, is highly conserved. And here's again what we saw. So if we addressing the hypothesis of sequence heterochrony in the ancestral, which was the red jungle fowl, in the descendant breeds, we did see a little bit of shifting, but not a whole lot. And then the next thing that we wanted to test was if there were changes in the allometry of traits, in the growth rates of individual traits. And so in panel B, we're showing some of the hypothetical scenarios for how that could happen. We have in red, alpha ancestor, showing the relationship between trait size and developmental time. And then we see shifts in the initial parameters in descendants. And then we also see shifts in the slopes. So the rates can change. So we're showing here positive allometry and negative allometry. So reductions in trait size, of course, should be produced by changes in by the slowdown of growth rates of traits. And so so in the same study, we measured a few traits: upper jaw length, lower jaw length, wing length, and also eye length. And so we did find some differences that were perhaps not so surprising, because it turns out that the broiler chicken that's been bred for meat production has undergone a major reduction in the wings. And that was interesting to find, because we proposed that that's likely because a lot of tissue resources are redirected to the production of hypertrophy breast muscles in that breed. And then we also found some interesting differences in the white leghorn egg breed that we could not really link to egg production per se, but we think that there are major differences that are most likely correlated effects of selection. And so the main findings or the main conclusions from the study were that exaggerated traits most likely or can be produced by basically selecting for shifts in allometric relationships in chicken. But generally speaking, those crucial developmental events that happen earlier in development are conserved. We also did a much broader analysis. We did a follow-up study to see what was happening in the rest of birds. What we're looking at here in this heat map visualization is the timing for about 115 developmental characters in birds. And they're arranged according to phylogeny. Excuse me. And there is not a whole lot of information in most bird species, actually. Birds have about 10,000 species, and we're only showing here about 20 to 25. So most of what we know about bird development comes from chicken and other domesticated breeds, including geese. And but we were able to extract data from the literature. I should say that this is all data from the literature for some interesting species. We found some interesting things, but what we wanted to know was what happens if we compare to our domesticated species, our model species, which is chicken. And then we also find very strong congruence when we compare, for example, quail, which is another popular domesticated breed, and also used sometimes in developmental biology, as well as duck, which is in panel B. Quail is in panel A. And panel C, we compare to the domestic goose. And overall, we see a lot of congruence, a few shifts that we cannot really explain, and we think it's mainly due to sampling error. And so overall, what we conclude from our comparative embryological studies is that developmental sequences, especially the ones that give rise to the organism, those that control organogenesis, early pattern formation, and morphogenesis are going to be very conserved, highly evolutionarily conserved. And one prediction that we can make based on our studies is that many phenotypes are likely to arise by changes in allometric growth trajectories late in development, even after embryonic development. And so I think this is the end for this part of the talk. Now I'm going to move on to body size evolution and limb length in lizards. We performed a series of comparative phylogenetic studies. And the idea was to try to link patterns that we saw in evolution, such as in body size, to life history and development. And so in this study, we were interested in the evolution of body size, and there are two there are two main patterns by which body size evolution may arise. In panel A, we're looking at one where like it arises abruptly in a clade, and then another one in panel B where it's more of a gradual trend towards miniaturization, so decrease in body size. And so to really understand how that could happen, you have to know something about the starting parameters in development. So initial egg size, shown in panel C, or even blastomer number in some cases. So the initial number of cells earlier in development can also influence body size in embryos. And so to achieve a smaller embryo, you need to modify initial egg size as a starting condition. If you want to get, for example, a smaller pup or hatchling, then you need to modify how much energy can be converted to tissue, can be directed to tissue production. And the energy is usually allocated via an egg yolk or directly by the mother. And then also adult body size is usually determined by growth rate after embryonic development and also the acquisition of nutrients. So we wanted to test these three conditions but also we were interested in what I spoke about earlier, allometric trajectories and the different scenarios by which they can arise. And so on the left side, we're looking at one scenario where like body size increases rapidly, but then there's some early cessation or precocious cessation of the growth period relative to the ancestor, pictured here in blue. Or you can have situations where like the growth rate decreases dramatically, but the growth period itself is still the same as in the ancestor. So there are many ways by which adult body size can be determined in development. And then of course, what's happening in terms of size is not mutually exclusive from developmental processes. So maturation, skeletal maturation processes, muscle development, and many other processes depend on body size. And on the right hand side, we're looking at a scenario where, for example, a hypothetical developmental process might cease earlier relative to the ancestor, but the rate at which, for example, the rate of bone ossification, which is an example that I will show later, or any other developmental process might actually be elevated in a miniaturized species. And so to test these scenarios, I decided to focus on body size evolution in turtles. So the largest turtles that can be presently found are marine turtles. So the largest living turtles are marine turtles. And they're related to a clade of freshwater species from the New World, and that have undergone body size reduction gradually. And so that's the hypothesis that we wanted to test. And so to do that, we needed body size information for, in this panel A, I'm showing body size measured as carapace length for all turtle species. And then we zoomed in into the Chilidro clade, which is the freshwater species that are sister to marine turtles. So we simply did a ancestral state reconstruction, and we mapped the ancestral states onto the predicted ancestral states onto a phylogeny. And then if you look at the heat map visualization in the gray, in the gray box, there is a gradual shift towards a smaller body size, going from the earliest branching lineages to the later branching lineages in the tree, which is in the family or in the clade kind of Sternidae. And so we also in the C panel, we use fossils to calibrate, or I should not say to calibrate, but to guide the reconstruction of ancestral states. And in general, the prediction that there was a gradual decrease in body size with evolutionary time was supported. And they were looking at 95% confidence intervals of the predicted ancestral states at each node of the tree across the phylogeny. And so we have pretty strong support for the hypothesis that there was a gradual miniaturization in this clade. And so we actually then wanted to see what was happening in the natural populations of these species, which is the exciting part of the study, I think, because we went out and we collected eggs of some of these species. We also had a colleague who had been studying in the United States, in the middle part of the United States, had been studying some of these species for decades. And we used those data to just illustrate how growth rate has basically been dampened down a lot in our miniaturized species, represented here by yellow dots. So if we look at a plot of carapace length against years post-hatching, so if we're aging the turtles by the years after hatching, and then above the plot, you can see one of the larger size species, which I had to trap, and we collected eggs from them, and we took the eggs to the lab and study development. But this was really interesting to have these data and to do the work in the field. Then in the lab, of course, we described embryos, we incubated eggs. Again, you can split development into two main phases: organogenesis, which is where a lot of the differentiation, morphogenesis, and pattern formation occurs, and then growth. And then for this study, we were interested in what's happening in terms of cell division and proliferation during the growth phase of these species. Here we're looking at what we call stage 18. which is during that organogenesis period, and the miniaturized Caenosteron suburbum species. And then we compare at stage 25, which is more or less near hatching, below to a representative embryos of the much larger relative Chilydra serpentina. And so in this study, we compared life history parameters such as the amount of energy that was allocated to the species, which is represented by yolk size, and then and that can also be represented by initial egg mass. So the smaller species have smaller eggs, and the consequence of that is that you have less energy to sustain growth, and so smaller species have smaller or lower relative growth rates, and that's what we're looking at in panel B here. And so how does that affect development? We again did another phylogenetic analysis looking at the correlation between the relative duration of organogenesis and the relative duration of the growth period across the species. It turns out that the small species have a much longer duration of growth, and that's because their relative growth rates is much lower than the largest species. And so here we're performing correlations against those growth periods in terms of relative to the entire incubation length against the size of the hatchlings. And then lastly, I spoke about developmental processes. So it turns out that the miniaturized species undergoes hyperossification. So they actually initiate bone development. Let's say here we're looking at, in red, we're looking at ossified tissue. And so it's used as a proxy for bone maturation or skeletal maturation. And so what we found was that the smaller species actually began to develop bone much earlier than the larger species. And so this was really interesting because the expectation was that everything should be downscaled proportionally, but that's not actually the case. Okay, so this is the end for this example that I have for you concerning body size. We supported the hypothesis of phyletic miniaturization in turtles. We looked at several life history parameters. We know that initial egg size will determine body size at embryo differentiation, so during organogenesis. And then also the amount of energy that's allocated to eggs will determine body size at hatchling. And then ultimately, growth rate will determine body size at the adult stage. And an important finding was that the effects of miniaturization are not systemic. So the downscaling of developmental processes will not be proportional across the different hierarchical levels of the organism. All right, so last example in reptile evolution, we wanted to look at limb length in our European lizards, specifically in the Lacertini clade, which is actually distributed from as far west as Madeira to as far east as Japan. And there are many species. We try to sample as many of them as we could. They are mainly generalists, high vegetation or low vegetation, or rocky specialists. And so we wanted to perform a study where we correlated limb length with ecological niche, and eventually study what was happening in the development of these species. I should say that in neotropical species, there's a very strong correlation between ecological niche and limb length. But in this temperate clade, we did not find that correlation. So we went out and we measured limb length, hind limb length and foreign limb length in as many species as we could in the clade. And we could not really correlate limb length with ecological niche. And so we concluded that the expectation, which was a very general expectation for lizards, that limb length should predict ecological niche and eventually performance of lizards, is not really generalizable to all clades, particularly in temperate species. And so we wanted to focus on on one genus, and it's one called Lacerta, and it's broadly distributed from Portugal all the way deep into Siberia, Russia, and Eurasia. And so we actually were working with a colleague in Ukraine who sampled some of these species in the wild and was able to obtain eggs and incubate them in the lab. And here we go. Again, we wanted to look at allometric changes and how allometric changes in development ultimately determine limb size. And limb size, of course, is important for locomotion and performance in these species. And so specifically what we wanted to test was whether these changes in the allometry of limb length originate during embryonic development, as has been shown in other DYSTRON species, or whether they arise in post-hatching development, which is the time when they would be functionally relevant. So very briefly, what we found was that the relationships were highly conserved between limb length and body size. But what we did find was that in our Lacertini species, are a low vegetation assertive viridis specialist, what we did find was that adult limb length is determined by a slight shift and a slight increase in the growth rate of the hind limb. And this is essentially when it becomes functionally relevant for the species, because there are low vegetation specialists that have been shown experimentally to depend on speed based on their hind limb length, but not the other species. And so that's the end for that example that I have for you, looking at microevolution. Now I'm going to talk about what's going on in the lab in our group at the moment. Most recently, we were looking at students in one of our courses in the master's program in evolutionary developmental biology. We're looking at the evolution of morphogenesis of the heart, which is a really interesting topic because there seems to be a gradient in terms of the numbers of chambers that are in the heart. And this, of course, affects the physiology and depends on the physiology of organisms. Reptiles have 3 chambers, while birds and mammals have 4. And we had some colleagues that were studying this because it's been predicted, actually, that mammals and birds have independently evolved a 4-chamber heart. But what was found recently, actually, is that mammals and birds do share a common developmental origin in terms of heart development, because their embryos actually mirror reptilian embryos during development, and that they will also have three chambers as embryos. But more importantly, perhaps, is that this condition of 3 versus 4 chambers of the heart is associated with a pathology in humans. It's really interesting that one of the most common congenital heart diseases in humans is caused by the failure of one of the walls of the heart to fuse. And so in congenital heart disease in some humans, you could claim actually that it's essentially a reptile-like phenotype. And so we were working with some colleagues from the Gladstone Institute in California, in the US, who gave us gene expression data to work with the students in the class. And we were recently looking at chicken embryonic development, sampling hearts, and we had already received gene expression data, which is what I will talk about in the slide here, for 3 stages of development in the heart of chicken. And we did a comparison of before septation, so before chamber differentiation and formation. So when the architecture of the heart was very raw, so to speak, and then that corresponds to stage 19 in chicken. And during the process of septation or heart chamber formation or differentiation, that corresponds to stage 25, more or less. And we did a comparison of what's happening in terms of gene expression. So I'm showing here something that's called a volcano plot with on the y-axis, essentially the significance of a contrast between those stages of development that I just mentioned. And then on the x-axis, we're looking at the direction and the strength of the differences, so the effect size, where we do pairwise comparisons of gene expression of the same gene across the stages of development. And so basically, to summarize what we found, that many genes involved in regeneration, so morphogenesis and pattern formation, were downregulated or expressed at lower levels when you go from before to during chamber formation. And then one interesting finding that we have, because for us, anything concerning the extracellular matrix is important in our research, is that tenacin, a very important gene involved in tissue remodeling, was expressed at a very different level. And so that was the end of our exercise. But we continue to do this type of work with skeletal muscle. And so skeletal muscle is the focus of the lab at the moment. Here we're looking at the anatomy of that. So there are several levels of organization. Here we're showing the outer layer of tissue that surrounds the inner layers of multiple fibers, including the perimysium and the endomysium. And eventually we can work all the way down to the raw building blocks of muscle, which is collagen. And I will show you more details of that here. So in our group, we're looking a lot at what I like to call the protein building blocks of muscle. And specifically, we are focusing right now on one called laminin, or there are several laminins. And this is important for us because it's involved in muscular dystrophies in mammalian systems. And we have a mouse model called the DYW model that's deficient in the gene LAMA2. And at the moment, we have several experiments in the lab looking at how this affects cell behavior, cell states during the onset of dystrophy or muscular dystrophy in mice. And so we know that happens at stage 18E18.5. And we're focusing on what's happening a little bit before that and also afterwards. And the cellular process that we're interested in concerns the division of stem cells, and also how myoblasts arise. So there's two ways in which that can happen. There's a differentiation, or basically a decision-making process that cells that are adjacent to the myofiber undergo. And so the current working hypothesis is that defects in our LAMA2, or the deficiency of LAMA2 in the basement membrane, also has effects on how stem cells will behave and ultimately produce myoblasts. And myoblasts, of course, are the cells that basically make up muscle fibers. And so this is critical for us because when this doesn't happen correctly, you get muscle weakness and reduced growth and malfunction altogether of the musculoskeletal system. And so that's the current focus of our lab. And my work so far has contributed to this by looking at gene expression profiles of before and during, let's say, the onset of muscular dystrophy in our DY2, in our DYW model. And so far, we did see a strong gene expression signal in terms of those extracellular matrix protein building blocks that I mentioned earlier. In terms of the genes that encode for those proteins, we are seeing a strong signature for a difference between our wild type and our DYW model. And as far as like what the cells are doing, this is something that we're still investigating. Our gene expression data so far did not yield a major difference in terms of the cell cycle genes, and this is because of technical reasons that I will explain next. We know that there's asymmetric cell division and symmetric cell division going on adjacent to the myofiber. And we previously focused on these populations. We previously focused on stem cells and myoblasts. And these cells are actually all at different phases of the life cycle of the cell cycle. And I like to call that proliferation and differentiation continuum. And so what we're proposing to do next is to employ single cell RNA-seq. And we're going to try to reconstruct gene profiles or gene expression profiles for individual cell types at individual within given proliferation and differentiation states. That's future work that we're hoping to do in the lab, but we're continuing to investigate this with a variety of molecular and cell biology techniques. And so, yeah, so I just want to close out this talk. I think the purpose of my talk was just to really highlight how developmental biology is relevant to evolution and biomedicine and many other applications. We know that different developmental processes, such as differentiation, pattern formation, and growth, can be viewed as a series of cell states. And they can be studied by via gene expression, which is my current specialty in the group. And ultimately, all of these processes do inform interesting patterns of evolution. And so I would like to end the talk with that and just say thanks to all my group members and all the people who helped out with the data that I showed you today. And now I think I have time for some talks. And I just wanted to say that we're in the C2 building. Stop by, or please feel free to contact me or anyone in the group if you have any questions about developmental biology or any kind of integrative question that you might have involving development. Thank you.
Thank you very much. Great talk, and we have time for questions, so I don't know who wants to start, but lots of clapping and congratulating you. So we have just to wait to see questions that people may have.
I have a question about the work on the limb length in lizards. Did you see any so you compared to body length, the limb length and body length, right? Mm-hmm. I'm not sure I got exactly, maybe you said the answer to this question, but I'm not sure I got it. So did you see any correlation, like longer bodies have shorter limbs, or there was no correlation in that sense?
A little bit of that. We saw a little bit of that with longer bodies tended to have longer hind limbs. Okay. In these species, and then also there was a genus of climbing lizards from Japan that have just very short hind limbs and forelimbs. So we saw really interesting patterns like this. Mm-hmm. And yeah, of course, many correlations, but earlier studies basically had painted a very simplified picture of like the ecology matches the limb length and so on. Yeah. But not in this case.
Yeah, actually, it's interesting because our professor Crespo, who worked on evolution and development, and he has now been retired for several years, he had this personal theory that the energy either went into body length or limb length. So he proposed, based on just not collecting data, but on correlation, that that the energy of making body length would mean that the animal would have less energy to make limbs. But I guess it's more complicated than that, because you also have the ecological pressures to. Okay, thank you.
We have one other question from Professora Margarida.
Hi, sorry, I was not able to attend completely because I'm moving the lab and there was an emergency, so I had to go when you were speaking about domestication, which is for me one of my most interesting topics. So I don't know if my question is out of place, but I was wondering, so when you consider the diversity of races, for instance, that you get, I was thinking about dogs, but also pigeons, for instance. There's also lots of crosses, right? So lots of hybrids that really involve very complicated genetic basis of the development. Have you considered also studying that, or that is not so interesting for you to study?
I think colleagues have before looked at that in chickens especially. So across the genomes, many SNPs have been found, many alleles have been found. The big challenge there is that oftentimes the alleles, it's very difficult to associate them to phenotypes, and in development you really have to know what's going on in terms of how these things influence regulation of genes, and so there's a huge gap there. But I think in the future, a lot of progress will be made in that. So finding those alleles like in chicken or pigeons, I think that has been done in pigeons a lot, but linking it to like what's happening in the embryo is very challenging at the moment.
And I was wondering that it may also involve like epistatic effects, right? When you cross different genetic backgrounds, and this may affect a lot also then the development of the different races, right? That might be also a challenge.
Yeah, epistatic effects. But ultimately, for breeding for agricultural purposes, for example, you're selecting for anything that will be viable. And so one kind of trivial conclusion from our study was that, of course, development will be conserved, because if it's not, then you're not going to have viable offspring.
Of course. No, of course.
Yeah.
Okay, thank you.
So we have Professor Gabriela.
I have a minute. Thank you very much, Antonio. That was really, really interesting. I wanted just to ask you something. I think you talked about the mid-developmental stage. And if I understood correctly, you said that it was very frequent to have a lot of mutations in that stage.
No, there was an experiment that induced many mutations during that stage. And so the prediction is that selection will not favor any mutation that.
Okay. That was my question, because you said it is very conserved, but then I didn't understand why then you had lots of mutations. Okay, perfect. Thank you very much.
So I don't know if there are any other questions. Just wait a bit. Of course, the talk will be available on YouTube. So if you have more questions to ask Antonio, I think you can, and then we will pass to Antonio, or you can ask him directly through the email we have in our website, in CE3C website. But I don't see any other hands. So I think we are finishing. Thank you very, very much, Antonio and Professor Salveig and all, for participating and for this discussion. I hope to see you next week in the next Encontro Ciência. And any questions you want to ask to Antonio, and you will be able to see later the talk in YouTube. I think we will have it today. So if you can ask him through the email. Thank you very much, and see you next time.
Thank you.
Thank you.